Hello friend, welcome once again. Today we will discuss the second part of JRF 2025 question paper. So let's start. The first one is based on the fat percentage. Arrange the following in descending order. So the maximum fat present in the walnut, followed by almond, then cashew nut, and last in avocado. This is very simple question. Another is the calculate the quantity of DAP in which 18 kg, 20 and 0 kg NAP is uh, NPK is present and urea has 46% nitrogen. To fulfill the recommended dose of 18 kg nitrogen, 20 kg phosphorus and 0 kg potassium in a vegetable crop. So this is a calculation types of question. So if you know the calculation, then it's okay. Otherwise I will let you know. First one is you how you calculate. First one is the quantity of fertilizer. Any fertilizer if you want to calculate, then the formula of that is quantity of that particular fertilizer and how much that fertilizer is required divided by the percentage of that much fertilizer. Means if the suppose you want to nitrogen from the urea, then the quantity of urea, how you calculate required urea divided by urea means nitrogen present in the urea into 100. This is the basic formula for the calculation. So let's we start. Then first we calculate the DAP. So the quantity is Q of DAP is equal to required P. Required means R P. Okay, divided by percent of P present in DAP. Okay, into 100. This is the formula. So if you calculate the quantity of DAP you want and the requirement of phosphorus. So how much you require? 20 kg. So here you keep the 20 divided by how much phosphorus present in the DAP. So here how much you have? 20. Then again you put here the 20. Okay, into 100. 20 20 cancel, you got the 100. So 100 kg DAP is required to fulfill the how much? 20 kg phosphorus. Then you calculate the nitrogen. So nitrogen from the DAP, because here the DAP also having the 18% nitrogen. So first you calculate the nitrogen percent from DAP, then we'll go for urea. So nitrogen from DAP, okay, is equal to in DAP how many percentage of nitrogen? 18%. So 18, and you apply 80 kg here, 100 kg of DAP. So you keep here 100 kg and into 100. 100 to 100 cancel, you got the 18 kg. Means if you are applying the 100 kg of DAP, you got the 18 kg of nitrogen from the DAP. Then you reduce the total amount of nitrogen from this DAP. So how much you required? 80 kg. So from 80 kg to minus 18 kg. Then you got how much? 62 kg. Then now you want to apply the 62 kg from the urea. Then you calculate the nitrogen from the urea. Okay, urea is U. Same formula. How much you want? 62 kg. This one is keep here, divided by how much quantity present of nitrogen present in the urea is 46%. You know, this is very simple. And then into 100. You calculate this and you got the 135 kg. Okay, so your answer is 100 kg DAP, this one, and 135 kg of urea. This is very simple question. Okay, इतना तो कैलकुलेशन आना चाहिए. ऐसे किसी भी फर्टिलाइजर का पूछा जा रहा है तो इस तरह से आप कैलकुलेट कर सकते हैं. Another one is the which one of the following line of onion has the true genetic constitution of N, M, small ms, small ms. So N and small ms present in the B line. Okay, and if the capital S and small ms same like small ms then this is A line. Okay, this one. Okay, if the capital N small ms then it is B line. Rest of another line is C line and D line तो होता नहीं. Okay, another is Jonathan spot is the physiological disorder of apple. Yes, it is true. Calcium deficiency is the sole cause of disease. No. Yes, it is due to calcium deficiency, but not only the calcium is responsible for this disorder. There are several factors, means that is basically related to post-harvest handling, such as delay cooling, high temperature, CO2 deficiency. Several factors are responsible for the Jonathan spot. So here the calcium is not only single responsible factor. Therefore, the R is correct and R is not correct and A is correct only. Okay, so you have third one is your answer. Cucurbits does not suffer much from inbreeding depression. Okay, it is very simple. Andromonoecious and hermaphrodite sex form favor a high degree of natural self-pollination. Yes, both are true. And due to presence of this two sex form, it having the higher self pollination. If the higher self pollination, then always the inbreeding depression is high. But in the case of cucurbit, the inbreeding depression is low. Why? Because their ancestor, basically the ancestor of almost all cucurbits, having the हर्माप्रोडाइट नेचर. अर्लियर वो जो भी इनके एंसेस्टर थे वो हर्माप्रोडाइट नेचर थे. उन्हीं से इवोल्यूशन के टाइम देयर आर सेवरल सेक्स फॉर्म, बहुत सारे सेक्स उसमें चेंजेज हुए. बट उसके जो पूर्वज थे, जो एंसेस्टर थे, वो हर्माप्रोडाइट थे. इस वजह से उसमें इनब्रीडिंग डिफरेंशन नहीं होता. ये इंपॉर्टेंट रीजन है. ओके, सो यू हैव फर्स्ट वन इज करेक्ट. Another is the mutation which occurs in one layer and along the side of apex is known as. So answering mericlinal chimera. There are three types of chimera. First one is basically first we lets you know there are three layers present in the plant, that is L1, L2 and L3. Okay. Basically it is divided in plant in three different layers. First one, second one, third one. First one is form the periderm of the plant. Okay, outer part. Second one is responsible for the gamete formation. If the mutation occur in the second layer, okay, means L2 layer, then it will segregate and go in the next generation. And if the mutation occur in the L1 layer or L3 layer only, then it not go in next generation. Okay. And third one is the form the cortex of the plant, that is L3 layer. And L1 and L2 is called tunica, and L3 is called carpus. Okay. तो मेरिक्नल में क्या होता है, if the meridional chimera are there, then mutation is occur in the first layer only, first layer, and only some part of this, okay, not the all part. That is called meridional chimera, and that is not stable, वो stable नहीं होता. Second one is the sectorial chimera. If the chimera is present in the means mutation present in the a sector, means इतने पार्ट में होगा, सेक्टर में होगा, पूरा पार्ट कवर नहीं करेगा, then it is called sectorial. Again it is not stable in nature. Okay, यह भी stable नहीं रहता. But if we took the some bud from these mutated part and propagated vegetatively, तो वो उस जो न्यू प्लांट्स हमारा जनरेट होगा उसमें वो म्यूटेशन दिखेगा. बट रेस्ट ऑफ पार्ट आर फ्री फ्रॉम द chimera. And third one is the most important, periclinal chimera. If the mutation is present in the whole layer, either it is this one and this one, and cover the entire plant part, okay, if the present in the full layer, then जो ये plant का diameter है चारों साइड से cover रहेगा, okay, तो वो कहलाता है हमारा पेरिक्लिनल कैमरा, और ये हाईली स्टेबल रहता है. ओके, पूरा जो लेयर रहेगा ये पूरा म्यूटेट होगा और ये कवर किए रहेगा इंटायर रेस्ट ऑफ अनदर लेयर्स. ओके, मोस्टली ये बी लेयर्स में रहता है, जो हमारा L2 लेयर्स है उसमें पाया जाता है पेरिंकिल में. सो दीज आर द बेसिक थिंग्स ऑफ रिगार्डिंग द काइमेरा. ओके, so basically this is a three types and three layers, L1, L2, L3, tunica and carpus. और इसमें सेल डिवीजन एंटीक्लिनल होता है, पेरिक्लिनल भी होता है, और इस टाइप से बहुत सारे इसमें कॉन्सेप्ट्स हैं. ओके, another is the glucosinolates have been proposed a chemical markers in chemotaxonomy of brassica vegetable. Okay, it is a true because in the brassica the glucosinolate is present, which is commercially used as a chemotaxonomy for the plant or species identification. Another is the region, the total amount and profile of glucosinolate vary among the brassica species. Yes, if it is a vary means the concentration differ from one genotype to another genotype, then we have only classified it according to their concentration and their composition. Okay, if the same concentration and composition is present in the all population, then no use for the no use for their marker or classification or chemotaxonomy. Okay, जब उसमें वेरिएशन रहेगा देन ओनली वी हैव द अपॉर्चुनिटी टू क्लासिफाई दिस. तो यहाँ पे दोनों ऑप्शन आपका करेक्ट है एंड आर इज करेक्ट एक्सप्लेनिंग टू ए. अनदर क्वेश्चन इज M1 capital R small m. These are the genetic constitution of the papaya and their character. If the M1 capital R small m is present, then it is the sex reversal but homozygous, because here the capital R present, then it is the homozygous condition. Okay, then you have the answer is sex reversal homozygous. Okay, but here the M1 capital R and small r means this is the heterozygous in nature, then the sex reversal in heterozygous male plant. Okay, this one is the M1 small r and small m, then it is pure male types, and this M2M you already know, this is the hermaphrodite type. So this is a very simple question, but must you know the what is the basic genetic constitution, how it is a heterozygous, how it is a homozygous, which is responsible for the male, which one is responsible for the female, and which one is responsible for hermaphrodite, which is the stable, which is mostly in sex reversal condition is present. Okay, carpilloidy, there are several things is important in the papaya section. Another question is which of the following method used for propagate the jasmine and hydrangea plant? So semi hardwood cutting. This is a simple question. Secure the cashew variety according to their parentage. This is an very बकवास क्वेश्चन है इस टाइप्स क्वेश्चन why they are asking I don't know but इस टाइप्स के इतना इम्पोर्टेंस में नहीं है तो no need to remember these types of parentage. Okay. Which of the following is considered as a progenitor species of loofah gourd? Loofah graveolens. This is a simple question. The differential growth response with greater response in the upper surface of organ is known as. If the growth response present in the upper surface of organ, this is sorry, then it is called epinesty. If the growth response present in the lower case of the organ, then it is called hyponesty. Basically these types are basically growth responses, how the growth is occur based on the different response. Next one is the nastic tropin. You know the several types of tropin: phototropin, geotropin, thermotropin. These are several types of growth behavior present in the plants. Accordingly, they have asked this question. Another is which of the following cultivars belong to cut standard types of chrysanthemum? So this is the chrysanthemum variety. You know to match according to the cut standard types. So Pusa centeni, Sadbhavana, you have Snowball, you have Thai Chain Queen. So A, C, and D are the cut standard types. Okay. Which of the following architect has given the concept of French style garden? So Le Notre is the father of French style garden. Another is the breeding method and its application. So first we need the application, then we predict which breeding method used for this. Okay, breeding method used to combine desirable trait from two different species. Okay, where प्रीजाइगोटिक बैरियर आर एग्जिस्ट मीन्स पॉलिनेशन के पहले ही उसमें बैरियर, फर्टिलाइजेशन के पहले ही बैरियर्स आ रहे हैं, देन विच ब्रीडिंग मेथड बी यूज? सो Markstein embryo rescue somatic hybrid. Here the somatic hybridization. In the somatic hybridization, we use the cytoplasm of both parent. हम दोनों पेरेंट्स के साइटोप्लाज्म को लेते हैं, उसको आर्टिफिशियली फ्यूज करते हैं, उसमें सेक्सुअल हाइब्रिडाइजेशन नहीं होता, हम उसको ए प्लस बी लिखते हैं ऐसे ए प्लस बी, नॉट ए इंटू बी. ओके, this is cytoplasmic hybrid, hybrid, bybrid, different kinds of hybrid will present. Okay, so is somatic hybrid commonly used for those crops where the pre-zygotic barrier exists, means totally incompatible है, pollination fertilization होगा नहीं, that condition में wide hybridization में use होगा उसको. Second one is the process of selecting and interbreeding, selecting then interbreeding plants ओवर सेवरल जनरेशन. सेलेक्ट करेंगे बट उसको सेवरल ईच ईयर में उसको इंटरब्रीड करेंगे वन बाई अनदर. तो ये हमारा जो रिकरेंट सिलेक्शन रहता है उसमें यूज होता है. रिकरेंट सिलेक्शन के आप मेथड उसका स्टेप देखेंगे तो उसमें सेम कंडीशन फॉलो होता है. ओके, नेक्स्ट वन इज टेक्निक यूज टू सक्सीड इंटर स्पेसिफिक हाइब्रिड. नॉर्मली जो इंटर स्पेसिफिक हाइब्रिड होता है जिसमें पोस्ट जाइगोटिक बैरियर्स रहता है. फर्स्ट वन है, दिस वन इज द प्री जाइगोटिक बैरियर, बट हेयर द पोस्ट जाइगोटिक बैरियर. इफ द पोस्ट जाइगोटिक बैरियर्स आर प्रेजेंट देन वी डू द एम्ब्रियो रेस्क्यू. मीन्स जो उसका बैरियर्स है पोस्ट जाओ इनकम्पेटिबिलिटी आता है, उसमें इनिशियल स्टेज में फर्टिलाइजेशन होता है, एम्ब्रियो फॉर्मेशन होता है, बट लेटर स्टेज में एम्ब्रियो जो है एबॉर्ट कर जाता है. तो हम क्या करते हैं, एबॉर्ट होने से पहले ही एम्ब्रियो को रेस्क्यू करते हैं और उसको इन वाइट्रो कंडीशन में ग्रो कर देते हैं. दैट इज कॉल्ड एम्ब्रियो रेस्क्यू, कमर्शियली यूज फॉर द इंटर स्पेसिफिक हाइब्रिडाइजेशन फॉर द overcome the post zygotic barriers. Okay, last one is the method involves selecting based on the molecular marker. So if the molecular markers means genes is present then marker resistant selection. Okay, Next question is, self incompatibility was first reported by Curator, and this case pollen grains fall to germinate on the stigma of the flower that produce them. Okay. Another is, in primula, pin flower have long styles. Yes, it is true. And short stamen. And thrum has the just opposite of this, have the short style and long stamens. Means pin having long style, pin to long, and thrum having short. You just remember the style, no need to remember the stamen. If the style is this, then opposite of that is stamen. दोनों में अपोजिट डायरेक्शन रहेगा, तो आपको यहाँ एक चीज का याद, इफ यदि पिन की बात की जाए, पिन मीन्स लॉन्ग रहेगा, तो यहाँ लॉन्ग स्टाइल, एंड थ्रम मीन्स शॉर्ट स्टाइल. इससे आप इजिली से इसको रिमेम्बर रख सकते हैं अपने माइंड में. ओके, तो यहाँ पे आपका दोनों स्टेटमेंट्स ट्रू है. ओके. Next one is, in papaya, pericarp of fruit composed of latissiferous develops close to. जो इसमें लैटेक्स निकलता है वो किससे क्लोज रहता है? जो इसका वास्कुलर बंडल है जाइलम फ्लोएम, उससे अटैच रहता है, इसलिए उसको फ्लो होता रहता है. ओके, वास्कुलर बंडल से. ओके. Which of the following pulse craft falls under the category of inflated pod? Not, they are not talking the flatted, means flatted and inflated. So if you see the fruits of Cajanus cajans, means pigeon pea, Vigna radiata, मूंग, Vigna mungo, black gram, इसके आप देखेंगे तो elongated and फ्लैट टाइप्स के इसके पॉट्स रहते हैं. बट इफ यू सी द सेसर अरेटिनम, मीन्स चना जो रहते हैं, चने के जो रहते हैं राउंड रहता है, इसका फ्लैट टाइप्स के पॉड नहीं रहते. सो बेस्ड ऑन मॉर्फोलॉजिकल you easily solve these types of questions. Okay. Which of the following variety are hybrid can produce the seeds in subtropical climate? So this is the Pusa mukta cabbage. These are the cabbage variety, and you know the Pusa ageti is the early and tropical season cabbage that will able to produce seed under the plant's condition, and rest of another types which having the typically temperate types will produce only fruits in the temperate region where the vernalization requirement is fulfilled totally. Okay. Next one is the ideal sucker size of planting of pineapple. So pineapple, this is a simple question. Sucker size is 450 and slip size is 350. This is the slip size, and the sucker size is 450 gram. This one is the 450 gram suckers. Okay. Very simple question and repeated question also. Next one, the zinc deficiency in apple is commonly called russet. Okay, russet is called in not only the apple, almost all crops. Okay, जहाँ पे जिंक डिफिशियंसी रहता है उसमें डॉर्फ एंड स्टेंटेड ग्रोथ होता है, जिसको हम रोजेट बोलते हैं. Next one is, rosette usually first appears on the summer season. First appears. Okay, first appears means when it will show the first symptom. जब आफ्टर प्रूनिंग के टाइम से जब डार्मेंसी ब्रेक होता है, स्प्राउटिंग के टाइम पे ही इसका जो है सिम्टम्स दिखता है. सो नॉट द समर सीजन व्हेन द स्प्राउटिंग इज इमर्ज, मीन्स इनिशिएट, दैट इज द बेसिकली स्प्रिंग सीजन, नॉट द समर सीजन. ओके, सो हियर द ड्यू टू समर सीजन the second statement is wrong, first statement is true. Next question is, there are several statements. Androgenesis is most commonly used onion for hybrid production. Commonly, they are talking the commonly means commonly regularly. तो regularly इसमें hybrid products होता है, तो एंड्रोजेनेसिस कॉमनली यूज नहीं होता. सेन H3 is the emerging player of haploid production technology. Yes, sen H3 is commonly used for the haploid production. Basically, what is the sen H3? Centromeric histone H3. This is the centromeric histone H3. What is the role of this? Recognize where the centromere should attach to spindle fiber. When the जब इसमें जो हमारा माइटोसिस या मियोसिस होता है, तो ये रिकॉग्नाइज करता है स्पिंडल फाइबर को कहाँ पे अटैच करना है। जो इसका सेंट्रोमेरिक रीजन रहता है वहाँ पे ये CNH3 प्रेजेंट रहता है, वहाँ पे ये रिकॉग्नाइज करता है कि यहाँ पे आपको अटैच करना है। एंड what is the basic step how the haploid is produced? If the correct first step was step first one is the correct the plant with the modified CNH3. The plant first plant, this is the plant first in it having the modified CNH3 protein, means this is a types of proteins okay present, and एक में जो है modified types के। सेकंड स्टेप क्रॉस इट विथ नॉर्मल, सेकंड पेरेंट P2 पेरेंट इज नॉर्मल okay, this one is the normal and this one is the mutated type means modified CNH3. Then what will happen during the embryo development? Chromosome will defective CNH3, जो इसका डिफेक्टिव्स है, जो CNH3 है, इसका क्या करेगा, क्रोमोसोम विद डिफेक्ट हो जाएगा, वो एलिमिनेट हो जाएगा, डिग्रेड हो जाएगा, सिंपल टाइम में डिग्रेड हो जाएगा। तो हमारा क्या है इसमें जीनोम बचेगा, क्रोमोसोम बचेगा, सिर्फ पेरेंट 2 का बनेगा। जब माइटोसिस के टाइम से इसमें जो जो भी हमारा ये क्रोमोसोम बायोलेंट कंडीशन में हम बनाते हैं, थोड़ा इसके सही नहीं है, दिस कंडीशन में सेकंड में जब ये अटैच नहीं होता स्पेंडल फाइबर्स, तो सिंगल पार्ट है, suppose this one is degrade and you got the this one is, that is सिंगल पेरेंट करेगा, सिंगल पेरेंट करेगा तो होमोजाइगस रहेगा, मीन्स हेप्लॉइड कंडीशन में रहेगा। तो थर्ड स्टेप जो आपका फोर्थ स्टेप रहेगा उसमें से जो सिंगल जीनोम है, मीन्स यहाँ पे अगर फीमेल इसको यूज किए हैं तो ये रेस्ट फीमेल का पार्ट डिग्रेड हो गया, आपके पास मेल का जो जीनोम है यही रहेगा जो हैप्लॉइड रहेगा। तो हम हैप्लॉइड प्रोड्यूस करेंगे, फिर उसको डबल डबल हैप्लॉइड खालचिसिन ट्रीटमेंट करके फिर हम होमोजाइगोसिटी सिंगल जनरेशन में अचीव कर पाएंगे। सो दिस इज द CNH3 मैकेनिज्म यूज फॉर द हैप्लॉइड प्रोडक्शन ओके। Another option is a high cytokinin to arginine ratio is typically required to induce the shoot organogenesis. Okay, if you have the high cytokinin and low arginine, then the shoot organogenesis occur. And if you have the high arginine and low cytokinin, if their concentration is reverse, if the vice versa is condition is there, then what will happen, the root organogenesis, the cytokinin for shoot and arginine for the root, ऐसे आप याद रखेंगे। जब कंसंट्रेशन आर्जिन का ज्यादा रहेगा तो रूट इनिशिएट होगा, इफ द साइटोकाइनिन कंसंट्रेशन इज हायर देन द शूट इनिशिएशन विल अकर ओके। So next option is RAPT is a co-dominant molecular marker. No, this is wrong. Basically, RFLP, SSR, SNP, these are the co-dominant marker. What is the co-dominant marker and dominant marker? Then basically there are two types of marker. First one is the dominant marker, which cannot be able to differentiate the homozygous versus heterozygous. Means अगर आपके पास जीनोम है कैपिटल A एंड स्मॉल a, तो जो हमारा डोमिनेंट टाइप्स के रहेंगे वो इसको डिफरेंशिएट नहीं कर पाएंगे, कौन से में ए कैपिटल ए है, कौन से में स्मॉल ए है। ये डिफरेंशिएट करेगा कैपिटल ए को और बी को, इस टाइप्स का जो है, या फिर किसी में कैपिटल ए सेपरेट्स रहेंगे, किसी में स्मॉल ए सेपरेट्स रहेंगे, इस टाइप्स के जीनोम को डिफरेंशिएट करेगा। बट if you have the co-dominant marker, then it easily differentiate capital A types and capital A small a types, means जो हेट्रोजाइगस कंडीशन में रहेंगे उसको भी ये डिफरेंशिएट करते हैं, तो वो को डोमिनेंट नेचर के रहते हैं। तो दिस इज द टू टाइप्स, डोमिनेंट एंड को डोमिनेंट नेचर। तो आपको इसके एग्जांपल रिपीटेड एवरी ईयर पूछे जाते रहते हैं, तो आपको याद रखना है। Second question is molecular. So again is the molecular question. So molecular marker have been widely used in the barcoding of species. Yes. Genetic engineering and fingerprinting. Yes, it is also used. Identification of duplicates. Yes, based on the recombination means capital A, small a, their allele size. There are several methods which are used to differentiate duplicates. Even the minor differences is there. If the single point mutation are there, even though molecular marker based on the genome sequences are the flanking marker, they are easily differentiated with each other. Okay. Another is the selecting the core collection. Yes, these are the four steps which are used for the molecular marker. So all are the correct. So you have the third one is the correct option. Another is the climbers and their scientific name. So the coral vine, that is the Antigonon leptopus. So the special character of this Antigonon is the pink color flower. Okay, pink flower. So pink flower. Another is the trumpet climber, that is the Campsis grandiflora. This is the orange color flowers. Indian evi, Ficus repens. This is the most important climber. Basically, this is the used for the screening of walls, and also Indian originated, and you having the foliage plant, not the flower plant. This is the foliage plant. There are several characters. First one is the foliage plant, originated in India, means Indian origin. स्क्रीनिंग ऑफ वॉल, जो भी दीवार रहते हैं उस पे इसको स्क्रीनिंग में उस पे चढ़ा के इसको एज ए क्लिम्ब करके पूरे को ग्रीनरी बनाते हैं, तो स्क्रीनिंग पर्पज के लिए यूज होते हैं। तो इस वजह से बहुत इंपॉर्टेंट क्वेश्चन, बहुत बार ये पूछा जाता है। एंड पर्पल ब्रेथ इज पेट्रिया वेलुबलिस, पेट्रिया हैविंग द blue color flower, belong to verbenaceae family, and also used for the bonsai making. So it having also several character. Blue flowers, belong to verbenaceae family, star shaped flower, and used for the bonsai making. Basically what they will ask, they have asked जो भी important क्लिम्बर पेरिनियल ट्री एनिमल जो भी कुछ उसके स्पेशल कैरेक्टर्स हैं या स्पेशल नेम्स में भी अगर स्पेशलिटी है कुछ, उसके मैक्सिमम आई हैव ऑलरेडी डिस्कस, जिसके पिंक फ्लावर्स हैं या ब्लू फ्लावर्स हैं या फिर येलो फ्लावर्स के, उन टाइप्स के फ्लावर्स को मैक्सिमम ये टारगेट करते हैं एग्जाम में। तो आपको इस टाइप्स के क्वेश्चन को याद रखना है। और एग्जाम इसका साइंटिफिक नेम, दिस इज द कॉमनली यूज्ड क्लिम्बर इन द आवर गार्डन। तो इस टाइप से अगर आइडेंटिफिकेशन आपका अच्छा है तो भी आप इजीली से इसको सॉल्व कर सकते हैं। दिस इज नॉट ए टफ क्वेश्चन। ओके। Another is the in pineapple fruit weight increase with increased number of suckers per plant. This is a very simple and repeated question. Wherein the increase the number of slips delay the maturity. Yes, both statements are true. Therefore, in the case of pineapple, desuckering not commonly practiced, and the commercially one plant will produce only one. In the rare some condition they produce the two suckers. Therefore very limited suckers are there. Therefore commercially slips are used for the planting of pineapple. And also in opposite condition जहाँ पे suckers का production बहुत कम हो रहा है, if the large number of slips, then the delay and reduce fruit size and delay maturity. और slip जो रहता है बहुत ज़्यादा है, 3 5, even some plants having the maximum slips बहुत ज़्यादा प्रोड्यूस होते हैं। तो दोनों में अपोजिट कोरिलेशन रहता है, सकर्स में स्लिप्स में। रेस्ट ऑफ अनदर क्रॉप्स वेयर द सकर्स इज प्रेजेंट, वहां पे जितना सकर्स रहेगा उतना मदर प्लांट्स में प्लांट की क्वालिटी फ्रूट साइज वो रिड्यूस होगी। बट पाइनएप्पल में अपोजिट इसका होता है। इसमें जो सकर्स रहता है वो कंट्रीब्यूट करता है एनर्जी या फिर सोर्स को, फोटोसिंथेट को, मदर प्लांट को कंट्रीब्यूट करता है उसकी फ्रूट साइज के डेवलपमेंट को। तो अगर जितने ज़्यादा सकर्स रहेंगे प्लांट में उतना बिग फ्रूट और बेस्ट क्वालिटी फ्रूट हमें मिलेगा।